SwayamPrabha. Digital India. Educated India.
Biodegradation of textile effluent, particularly dyes. Lecture number 19. Let us now dwell a little deeper into how the biodegradation of dyes take place which are present in the textile effluent. Biodegradation of textile effluents, particularly with perspective to dyes. Biodegradation of textile effluents, particularly concerning dyes, is a crucial environmental concern due to their potential harm to the ecosystem and human health. Textile dyes are often complex organic molecules that can persist in the environment for long periods if not properly treated. Here is a brief overview of biodegradation process of textile dyes. Although we have studied this fleetingly in the previous few lectures, now we will see the mechanism by which the degradation actually takes place. Microbial degradation. Many textile dyes can be broken down by microorganisms such as bacteria and fungi. These microorganisms produce enzymes that can catalyze the breakdown of complex dye molecules into simpler, less harmful compounds. This process typically occurs under aerobic, which is with oxygen, conditions, although some anaerobic without oxygen degradation pathways also exist. Enzymatic action. Enzymes secreted by microorganisms play a crucial role in the biodegradation of textile dyes. These enzymes, known as oxidoridases, reductases, hydrolases, peroxidases, target specific chemical bonds within the dye molecule, facilitating their breakdown into smaller, more easily metabolizable fragments. Metabolic pathway. Microorganisms utilize various metabolic pathways to metabolize dye compounds. These pathways may involve sequential steps of oxidation, reduction, hydrolysis, and conjugation reactions, ultimately leading to the complete mineralization of dye molecules to carbon dioxide, water, and other simple inorganic compounds. Factors affecting biodegradation. Several factors influence the biodegradation of textile dyes, including pH, temperature, oxygen availability, nutrient levels, and the presence of inhibitory substances. Optimal conditions for microbial activity must be maintained to ensure efficient dye degradation. So, there are several factors which govern the degradation process. It is not that we can do the degradation by microbes at any and every temperature and pH. So, this has to be kept in mind. Microorganisms utilize various metabolic pathways to degrade organic pollutants present in textile effluent. These pathways involve a series of enzymatic reactions that break down complex organic molecules into simpler compounds, ultimately converting them into biomass, carbon dioxide, water, and other end products. Here are some of the key metabolic pathways employed by microorganisms in the degradation of textile dyes, which are present in the textile effluent. Aromatic compound degradation. Many organic pollutants found in textile effluent are aromatic compounds such as dyes and aromatic hydrocarbons. Microorganisms employ several metabolic pathways to degrade aromatic compounds, and this includes aerobic degradation. In aerobic degradation, the condition is such that microorganisms utilize oxygen as a terminal electron acceptor to oxidize aromatic compounds. This process typically involves the initial activation of aromatic ring by dioxygenases or monooxygenases, followed by ring cleavage reaction mediated by dioxygenases or extradiol cleavage enzymes. The resulting intermediates are further metabolized through central metabolic pathway such as the tricarboxylic acid TCA cycle or beta keto adipate pathway. So, these metabolic pathways are very well defined and very well understood. Anaerobic degradation. Under anaerobic degradation means it is devoid of oxygen. Certain microorganisms can degrade aromatic compounds using alternative electron acceptor such as nitrate, sulfate or carbon dioxide. Anaerobic degradation pathways include reductive dehalogenation, fermentative degradation and anaerobic ring cleavage reactions mediated by enzymes like reductive dehalogenases and benzoyl CoA reductases. Aliphatic compound degradation. Aliphatic compounds including alkanes, alcohols and fatty acids are also present in textile effluents. Microorganisms metabolize these compounds through various pathways. Alkane degradation. Alkanes are degraded by aerobic bacteria through the initial activation of CH bonds by alkane monooxygenases, followed by oxidation reaction catalyzed by alcohol and aldehyde dehydrogenases. This process generates fatty acid that can be further metabolized via beta oxidation pathways. So, you see that each of these pathways are quite different. The point of attack is also very different for the microbes. They will sometime attack through oxygen, without oxygen, or they will attack the CH bond. Fatty acid degradation. Fatty acids are degraded via beta oxidation pathways, where successive cycle of oxidation, hydration and cleavage lead to the release of acetyl CoA units that enter the TCA cycle for further oxidation and energy production. Nitrogen and sulfur metabolism. Microorganisms also play a role in the metabolism of nitrogen and sulfur compounds present in the textile effluent, including nitrates, nitrites, ammonia, sulfates and sulfides. These compounds are cycled through various metabolic pathways including denitrification, nitrification, ammonification and sulfate reduction, ultimately leading to the conversion of nitrogen and sulfur species into less harmful forms. So, basically what these microbes do is that they mineralize them or they break down the complex molecules into simpler molecules which are not so harmful. Carbon and energy metabolism. Microorganisms derive carbon and energy from organic pollutants present in the textile effluents through catabolic pathway such as glycolysis, the TCA cycle and oxidative phosphorylation. These metabolic pathways enable microorganisms to utilize organic carbon sources for growth and maintenance while producing ATP as an energy source. Overall, if we have to say, the metabolic versatility of microorganisms enable them to degrade a wide range of organic pollutants present in the textile effluents through diverse enzymatic pathways. So, it is not just one pathway. There are several pathways which we have just learnt in the last few slides. Understanding these metabolic pathways is essential for developing effective bioremediation strategies for the treatment of textile wastewater. So, what it means is that if we do not understand the mechanism, how can we decide which bioremediation is apt for what removal? Like there are so many organic pollutants. Now, which one is preferably metabolized by microorganism, that understanding we should definitely have before proceeding. Bioremediation techniques. Bioremediation techniques such as activated sludge treatment, biofiltration and constructed wetlands harness microbial activity to degrade textile dyes in wastewater treatment plants. And this is a very known fact. We have learned this in several lectures. These techniques can be effective in removing a wide range of dye pollutants from wastewater, although the efficiency may vary depending on the specific dye and microbial community present. So, some, as we had learned sometimes back, that some microbes are very specialized. They target only special molecules. So, suppose if there is a surfactant molecule and there is a species of microbe which is only acting upon the surfactant, then that effectively will be far more, instead of using a consortia of microbes which will not target so effectively. Challenges and future directions. Despite significant progress in bioremediation research, challenges remain, including the degradation of complex dye mixtures, the identification of novel dye degrading microbes, and the development of cost effective and sustainable bioremediation strategies. Future research efforts are focused on exploring microbial consortia, genetic engineering of dye degrading enzymes, and integrating biodegradation processes into sustainable wastewater treatment system. So, still there is lot of research that needs to be done to be understood what works for what, which microbe is best for metabolizing which compound. Overall, biodegradation offers a promising approach for mitigating the environmental impact of textile dyeing processes. But continued innovation and collaboration are still needed to address remaining challenges and develop effective solutions. Microbial degradation of textile effluent involves the breakdown of organic compounds present in the wastewater by microorganisms such as bacteria, fungi, algae, protozoa. Textile effluents often contain a diverse range of organic pollutants including dyes, solvents, surfactants, emulsifier and other chemicals used in the textile industry. Microbial degradation plays a crucial role in the treatment of textile wastewater, contributing to the removal or transformation of these pollutants into less harmful or non-toxic forms. Here is how microbial degradation typically occurs in textile effluent treatment. So, what happens exactly during the aerobic biodegradation? In aerobic condition, microorganisms utilize oxygen because it is in the presence of oxygen to metabolize organic pollutants present in the textile effluent. This process involves the enzymatic breakdown of complex organic molecules into simpler molecules such as carbon dioxide, water and microbial biomass. Aerobic degradation is typically more efficient for readily biodegradable compound such as simple organic dyes and surfactant. So, this will work only on pollutants, organic pollutants which are easily biodegradable, which means that they can be simply attacked by oxygen, which will initiate the metabolic reaction of the enzymes present in the microbes and the degradation will happen. Anaerobic biodegradation. Anaerobic degradation occurs in the absence of oxygen and is often employed in conjunction with aerobic treatment methods. Certain microorganisms known as anaerobes can metabolize organic pollutants using alternative electron acceptors such as nitrate, sulfate or carbon dioxide. Anaerobic digestion processes such as anaerobic lagoons or biogas reactors can be used to treat textile effluent and produce biogas as a byproduct. So, this we have learnt in earlier lectures as well. But what is the mechanism? How does the microbe exude enzyme? What are the types of enzymes that are exuded which act upon the organic pollutant? Enzymatic action. Microbial degradation of textile effluent relies on the activity of various enzymes produced by the microorganisms. These enzymes including oxidases, hydrolases and peroxidases target specific chemical bonds within organic pollutants, facilitating their breakdown into smaller, more biodegradable fragments. Enzymatic degradation pathway can vary depending on the structure and composition of the pollutant present in the effluent. So, enzymatic action also is targeted. It chooses a specific pollutant and acts upon it. It is not a generalized process that any time any enzyme will work on any substrate. It is more like a lock and key arrangement. Just the way only one key can open the lock, similarly one enzyme can degrade one type of compound. Bioremediation techniques. Bioremediation techniques harness microbial degradation processes to treat textile effluent effectively. This we have understood by now. Common bioremediation techniques include activated sludge system, biological aerated filters, sequencing batch reactors and constructed wetland. We have studied this in detail. These systems provide favorable condition for microbial growth and activity, promoting the biodegradation of organic pollutants in wastewater. So, we have already understood that how in these reactors, that is activated sludge or trickling filters or constructed wetlands or sequencing batch reactor, the microbes are first developed, the cultures are grown and then the wastewater is run into it. Factors affecting biodegradation. Several factors influence the efficiency of microbial degradation in textile effluent treatment, including pH, temperature, nutrient availability, microbial diversity, and the concentration and complexity of the organic pollutants. So, there are several factors. It is not just if we maintain the pH, we will be able to do the biodegradation. If we maintain the temperature, we will be able to do the biodegradation. No, there is a whole lot of parameters that need to be monitored and maneuvered so that it is congenial for the microbes to grow in that. Once they grow, then the actual action begins. Optimal operating condition must be maintained to ensure the effective removal of contaminants and the stability of microbial communities within treatment system. Monitoring and optimization. Continuous monitoring and optimization of biodegradation processes are essential to achieve efficient treatment of textile effluent. Parameters such as pollutant removal efficiency, microbial biomass growth, and microbial community composition are monitored to assess treatment performance and identify opportunities for process optimization. Overall, if we have to look at the overview of microbial degradation, it plays a vital role in the treatment of textile effluent undoubtedly, offering a sustainable and environmentally friendly approach to mitigate the impact of textile industry wastewater on water resources and ecosystem, which has been the primary concern why we are learning this textile effluent topic at all. Continued research and innovation in bioremediation technologies are essential to address emerging pollutants and improve the efficiency and reliability of textile effluent treatment processes. So, therefore, it is very reliable, but we need to understand what are the parameters that need to be set, and only when we understand, then we can get the maximum benefit out of the microbial degradation. Enzymatic action. We have been saying, okay, it works on the enzymes, and the enzymes come from the microbes then react with the pollutant and so on. But what exactly happens? What is the chemistry behind it? Let us try to understand. Enzymatic action on textile effluent refers to the process by which enzymes catalyze the breakdown of complex organic compounds present in the wastewater generated by textile manufacturing processes. Enzymes are biological catalysts that accelerate chemical reactions, making them essential for the biodegradation and detoxification of pollutants in textile effluents. Here is how enzymatic action occurs in textile effluent treatment. Types of enzymes. Various types of enzymes play a role in the degradation of different classes of pollutants found in textile effluent. It is not just one enzyme, it is a consortia of enzyme which is given out by the microbe, and each of these enzymes work on particular organic compound, because as I told you, enzymes are catalysts and they are also having a lock and key kind of relationship with the substrate. These enzymes include oxidoreductases, hydrolases, peroxidases, esterases, among others. So, they belong to different classes of enzymes. Each enzyme targets specific chemical bonds within organic molecules, facilitating their conversion into simpler and more biodegradable compound. Oxidation reduction reactions. Oxidoreductase enzymes catalyze oxidation reduction or redox reactions, where electrons are transferred between molecules. These enzymes are involved in the oxidation of organic pollutants such as dyes and aromatic compounds to break them down into less toxic forms. For example, oxidoreductase includes lacases, peroxidases and dehydrogenases. So, within that broad class of oxidoreductase enzyme class, there are these lacases, peroxidases and dehydrogenases, which are individual classes of enzyme. Hydrolysis reaction. Hydrolase, as the name suggests, is an enzyme that catalyzes hydrolysis reaction, where chemical bonds are cleaved by the addition of water molecule. Hydrolases are effective in breaking down esters, ethers and other organic compounds present in the textile effluent. Lipases, proteases, celluloses are common hydrolases enzyme that can degrade lipids, proteins and cellulose respectively. So, you see, lipase cannot break down cellulose, protease cannot break down cellulose, protease only can break down protein, lipase can only break down lipids. So, there is a specificity in terms of the enzymes as well. Peroxidases action. Peroxidase enzyme such as horseradish peroxidase HRP catalyze the oxidation of organic substrates using hydrogen peroxide as a co-substrate. Peroxidases are capable of degrading a wide range of pollutants including phenolic compounds and aromatic amines. Although oxidative reactions are happening, including phenolic compound and aromatic amines through oxidative reactions. They play a crucial role in the detoxification of textile effluents containing azo-dyes and other aromatic pollutants. So, they are very, very specific for azo-dyes and aromatic pollutants. Esterase activity. Esterase enzyme catalyze the hydrolysis of ester bond present in the organic compound such as surfactants and emulsifiers used in the textile processing. By breaking down ester containing molecule, esterases contribute to the degradation of complex organic pollutants and the reduction of toxicity in textile effluent. So, each one is targeting only one type of compound. It is not a general reaction, it is not a general chemical reaction, but it is a targeted one-to-one identified mode of reaction. Enzyme optimization. Enzymatic degradation of textile effluents can be optimized by adjusting various factors including pH, temperature, substrate concentration and enzyme activity. Optimal condition for enzyme activity must be maintained to maximize the efficiency of pollutants degradation and minimize the formation of undesirable byproducts. Even here, just like chemical dosing, we were emphasizing about chemical management system in the traditional methods of mitigation of wastewater. Here also in enzyme degradation processes, all the parameters have to be kept in mind. It is not a general reaction. It is very specific reaction mode. Biotechnological application. Enzymes isolated from microorganisms or genetically engineered enzymes are increasingly being utilized in biotechnological applications for textile effluent treatment. Enzyme-based bioremediation processes offer advantages such as specificity, efficiency, and environmental compatibility compared to conventional chemical treatment methods. Enzymatic action of textile effluent. The enzymes used in the enzymatic bioremediation of textile industry effluent are mainly azoreductases, lacases, and peroxidases. Enzymatic bioremediation or even conventional treatment can generate byproducts that are equally toxic to the starting materials. Overall, enzymatic action plays a crucial role in the degradation and detoxification of organic pollutants in textile effluent, offering a promising approach for sustainable wastewater treatment in the textile industry. Continued research and development efforts are focused on identifying novel enzyme, optimizing enzyme-based processes, and integrating enzymatic treatments into comprehensive wastewater treatment strategies. Bioremediation techniques. Bioremediation techniques have been gaining increasing prominence worldwide due to high public acceptance, low cost compared to conventional remediation methods, high availability of enzymes, and minimal impact on the environment. So, because of the biotechnological development, the isolation of enzymes have become very easy, and that is why people have accepted this bioremediation technique using enzymes and using microbes as very readily available technology. It is low in cost as compared to the conventional remediation method, and there is no dearth of isolation of enzymes from the microbes. It is highly available. The exploration of enzyme for bioremediation has been of great interest due to their availability to function in wider range of pH and temperature in the presence of contaminants and saline concentration. So, more and more exploration is taking place for bioremediation methods where more versatility and more broad range enzyme reactions and working area of wide range pH and temperature are being worked on. Enzymatic bioremediation is an ecological, economical, promising and innovative technique. The process consists of exploring the typical characteristic of microorganisms or genetically modified organisms capable of producing specific enzyme to catalyze or metabolize the specific pollutant, transforming the toxic form into non-toxic form and sometimes into new products which are very safe. Enzymes can be used in free or immobilized form, the latter having the following advantages: long-term operational stability, easy recovery and reuse in the industrial application, which improves process performance and lowers overall cost. So, the immobilized form instead of the free form is very cheap, can be used again and again, and has a long term operational stability. That means enzyme do not get denatured at those temperatures and pH. Immobilization consists of coupling the enzyme with an insoluble support matrix to maintain an adequate geometry which guarantees greater stability to the enzyme. So, the beauty is that the enzyme is stuck on a support, and the support is designed in such a manner that it gives stability to the enzyme and does not let it get denatured. The bioremediation process using microbial enzyme can be slow. So far, only a few bacterial species have been able to produce enzymes with potent biodegradation capacity. So, usually these processes are slow. There are only a few cases where it is a very potent enzyme which acts upon very fast. Thus, the use of genetically modified organisms is more common due to their ability to produce large amounts of enzyme under optimized condition, and that is through biotechnological intervention. In the treatment of effluent from the textile industry, enzymes act on the dyes, generating precipitates that can be easily removed or chemically transformed into easy to treat compounds. The rate of dye degradation by enzymes will depend on the chemical structure of the dye, salt content, the concentration of metal ion, pH and temperature of the wastewater. So, the enzymes are very sensitive. Any change in pH, any change in temperature or chemical structure of the dye, if it is very complicated or if it has too much of salt content, can deter the degradation process. The enzymatic degradation of pollutants in textile effluent has several advantages such as specificity and selectivity to the substrate, in addition to being an accessible, efficient method that meets the principles of green chemistry. The requirement of large amounts of enzyme, high cost, thermal instability, inhibition of enzymatic activity, attack of certain enzymes by proteases and the formation of undesirable byproducts are the main difficulties or challenges related to the use of enzymatic degradation for wastewater. So, they have their own set of advantages as well as there are some drawbacks. Certain enzymes by proteases can form undesirable products, and that can be troubleshooting. Enzyme mediated bioremediation has gained notoriety due to its versatility and efficiency in the degradation of persistent organic pollutants, thus being applied in industrial, biotechnological and environmental processes. These enzymes can be obtained from the extraction of intracellular and extracellular metabolites from cultures of certain species of bacteria, fungi, algae and plants. Many of the tested can decolorize the dyes as well as provide a decrease in their toxicity, as in the case, for example, the horseradish peroxidase, which promotes the decrease of the toxicity of methyl orange dye. Dye decolorizing microorganisms produce a variety of enzymes including azoreductase, riboflavin reductase, lactase, peroxidases, NADH, DCIP reductase, tyrosinase reductase and aminopyrine N-demethylase, lignin peroxidase and veratril alcohol oxidase. Among those enzymes, The main ones responsible for decolorization of azo dyes are azoreductases, lacases and peroxidases, as what we learnt a while ago. Peroxidases play a role in degradation of azo dyes, and also oxidoreductases, which contain heme. Peroxidases are present in plants, microorganisms and animals. The mechanism of action of such enzyme is similar to that of lacases, providing the degradation of dye without the production of toxic byproducts. Peroxidases act specially on synthetic dyes, degrading their respective constituent through the oxidative polymerization of phenolic compounds to form insoluble polymers. An association between oxide reducing enzymes can significantly reduce the toxicity of the dyes. So, we know that at least by the end of this lecture you will understand that how the enzymatic action takes place. What are the main categories of enzyme that play a vital role in the microbial degradation. Moving on, surfactant based ionic liquids for degradation of dyes. There is another class of compounds which have now been used for the degradation of surfactants. A novel use of ionic liquids, ILS, with surfactant characteristic to improve the degradation of the largely used and highly hydrophobic textile dye indigo carmine by lacase. An initial screening on the activity of lacase in aqueous solution of 12 surfactant based ILS from 3 different families, namely tetraalkyl ammonium and imidazolium based cationic surfactants and collinium based anionic surfactants at different concentrations were carried out with decyl trimethyl ammonium bromide. Significant improvements in the activity of lacase was observed. These ILS were then investigated in aqueous solution to simultaneously encapsulate lacase and IC from the in situ enzymatic biodegradation of the dye. The use of ILS remarkably increases the degradation rate of the dye and decolorization efficiency. A degradation efficiency of IC of 82% is attained in just 5 minutes. Now, let us see how do these ionic liquids work in industry. When look at this chart, the effluent, when it has water and dye, and when more water and enzyme and liquid crystals are added into it, ionic crystals are added, then it gives water which can be reused back and enzymes like lacase plus surfactant based ionic liquids can actually work by biological activity and they can be maintained. So, what it means that it is a very safe method where there is 93% of dye degradation by the enzyme lacase in presence of surfactant based ionic liquids. So, it means that it actually amounts to a very safe method, and this was applied to decolorization of Remazol brilliant blue dye. The biotechnological potential of filamentous fungi from wastewater of industry, textile industry for the production of lacase, cellulase, amylase and lipase enzymes and their potential for discolorization capacity on Remazol brilliant blue R synthetic dye. The isolate aspergillus was presented in the best cellulase, amylase, lipase, lacase production as well as RBBR dye discoloration ability in solid medium, followed by isolate asparagillase able to synthesize cellulase, amylase and lacase, and had the capacity to discolor the 74.7% of RBBR, that is Remazole Brilliant Blue R synthetic dye in liquid medium. The results of this work encourage future studies of characterization, optimization, purification of the enzymes encountered, aiming to be used in bioremediation processes of textile industrial effluent. Filamentous fungi, just like liquid crystals, can also be ionic liquids, can be also very useful. Filamentous fungi used for textile wastewater treatment. Results obtained in the work demonstrated the biotechnological potential that filamentous fungi recovered from textile wastewater treatment plant samples may present. Fungi inhibiting this environment may produce several enzymes of industrial interest, including lipases, cellulases, amylases and lacases for use in the synthetic dye bleaching processes. That means the fungi that are already existing in the wastewater, they are inhabiting in that, and from that they are isolated, and enzymes are then produced from these fungi such as lipases, cellulases, amylases and lacases. And we have already seen that these enzymes are responsible for the biodegradation of certain compounds like dyes, surfactants and many other organic aromatic compounds. Further research is needed to reveal the importance of microbial communities present in industrial effluent samples and their dynamics within such harsh environment. The characterization of enzymes found using different microbial growth condition as well as to optimize, purify and apply them in pilot scale industrial processes will be the next stage of the work, as well as in situ bioremediation processes. So, there are several plans, like can it be generated in situ, need not be isolated first and then put into the reactor and so on and so forth, or can there be a way to purify these enzymes so that they are target specific and work very effectively in short period of time. Filamentous fungi for textile wastewater have been actually tried out. Filamentous fungi have harnessed attention to their potential in textile wastewater treatment due to their ability to degrade a wide range of organic pollutants, including dyes, surfactants and other textile industry related compounds. These fungi possess diverse enzymatic system that enable them to break down complex organic molecules, making them valuable candidates for bioremediation. Here is how filamentous fungi can be utilized for textile wastewater treatment. Lignolytic enzyme. Filamentous fungi such as white rot fungi produce lignolytic enzymes including licases, peroxidases and manganese peroxidase. These enzymes are capable of oxidizing a wide range of organic compounds including lignin, aromatic dyes and other recalcitrant pollutants present in the textile effluent. Lignolytic enzymes are particularly effective in degrading complex aromatic structures, breaking down chromophoric groups of the dyes, that is the color producing portion of the dye molecule, and facilitating their mineralization. So, what it means that these fungi have a different set of enzymes which work on different substrate, particularly lignin, which is very difficult to hydrolyze and break. They have lignolytic enzymes present in them. Decolorization of dyes. Filamentous fungi can efficiently decolorize textile dyes through enzymatic degradation and adsorption processes. Ligases in particular play a crucial role in the decolorization of azo, anthraquinone and triphenylmethane dyes by catalyzing the oxidation of their chromophoric groups. Additionally, fungi can produce extracellular polysaccharides and melamine-like pigments that bind and adsorb dye molecules, further contributing to decolorization. So, they work in two different sites. They are not only breaking the molecule, but they are also absorbing the molecule, and they produce extracellular polysaccharide and melamine-like pigments that bind and adsorb the dye. So, dual action is taking place. As a result, the decolorization seems to be very effective. Biodegradation of surfactant and textile auxiliary. Filamentous fungi possess versatile metabolic pathways that enable them to degrade surfactants, emulsifiers and other textile auxiliaries present in wastewater. Fungi produce a variety of extracellular enzymes including lipases, proteases and esterases, which catalyze the hydrolysis of ester, amide and other ether bonds present in the surfactant molecules. This enzymatic activity lead to the breakdown of the surfactant into simpler compounds that can be assimilated by fungi or further metabolized by microbial consortia. So, you see, they also work in unison with microbial consortia, but filamentous fungi by themselves do a lot. So, they have certain specific action role to play in the degradation of surfactants and other auxiliaries specifically. Tolerance to harsh condition. Many filamentous fungi exhibit robustness and adaptability to harsh environmental conditions commonly encountered in textile wastewater, including high salinity, alkalinity and presence of toxic metals and organic pollutants. Fungal mycelium forms a dense network of hyphae that enhances surface attachment and resistance to shear forces, facilitating the retention and degradation of pollutants in wastewater treatment system. So, they produce many hyphae, that means many branches, and on those branches the surface area increases and they are tolerant to all kinds of harsh condition. Biofilm formation and immobilization. Filamentous fungi have the ability to form biofilms on solid surfaces, including support materials in bioreactors and constructed wetland. Biofilm formation enhances the retention and activity of fungi in wastewater treatment system, improving the efficiency of pollutant removal. Additionally, fungal biomass can be immobilized onto inert carriers or substrate, allowing for the development of continuous flow bioreactors or decentralized wastewater treatment applications. Filamentous fungi overall offer promising opportunities for the bioremediation of textile wastewater due to their diverse enzymatic capabilities, robustness and adaptability to challenging environmental conditions. Further research is needed to optimize fungal based treatment systems and integrate them into sustainable wastewater management practices in the textile industry. To summarize, environmental impact, microbial diversity, enzymatic degradation. Textile dye pose significant environmental risk due to their persistent toxicity and potential contamination to the water bodies and soil. Biodegradation offers a sustainable approach to mitigate the environmental impact of the dyeing processes by transforming hazardous pollutants to non-toxic end product. Microorganisms play a central role in the biodegradation of textile dyes, utilizing diverse metabolic pathways and enzymatic systems to degrade a wide range of dyes molecules. Bacteria, fungi, algae have been identified as key biodegraders capable of metabolizing different classes of dyes under aerobic as well as anaerobic conditions. Enzymatic degradation. Enzymes produced by microorganisms such as oxidoreductases, hydrolases, peroxidases catalyze the breakdown of the dye molecule into smaller, more biodegradable fragments. These enzymes target specific chemical bonds within the dye structure, facilitating their degradation into non-toxic end products. To summarize, we can say that bioremediation technologies harness microbial degradation processes to treat textile effluent effectively. Activated sludge system, biofiltration, constructed wetland and enzymatic treatments are among the bioremediation technologies employed for the treatment of textile wastewater, offering cost effectiveness and environmental friendly solution. However, there are several challenges and there are future direction to work on significant areas. Future research efforts are focused on exploring microbial consortia, genetic engineering of dye degrading enzymes and integrating biodegradation system processes into sustainable wastewater treatment system. So, to conclude, the biodegradation of textile effluent, particularly dyes, is a multifaceted process that involves enzymatic action of microorganisms to break down complex organic pollutants into simpler and less harmful compounds. In conclusion, the biodegradation of textile effluent, particularly dyes, represent a promising approach to mitigate the environmental impact of textile dyeing processes and to ensure the sustainability of the textile industry. And we know that as long as the textile industries exist, we will have to look for newer and newer methods of mitigation of the pollution load. So, with this we have come to an end of this lecture. Thank you.